Okay, so first of all, good evening, everyone. First of all, I would like to thank the organizers for this very great two-day meeting. It's very interesting so far. I hope it'll still be like that. So as Mathilde said, I'm a postdoc in cell adhesion and mechanics lab in Institut Jacques Monod here in Paris, in the lab of Benoît Ladoux and René Marc-Mège. And we'll talk to you today about curvature-guided whole tissue epithelial rotation, as we call it, which is the project that I am doing here in the lab. And as you can already see in these two movies, basically we impose geometries in epithelial cells, MDCK cells mainly, and we can see how do they behave as single cells or collectively. So on the left, you can see some actin GFE cells, so we can look at actin, and here you see some histone GFE cells, so we can look at DNA and track the cells and so forth. So there are different examples of collective cell rotation that has been observed mainly in vitro in the past couple of years. So first, if you use some 2D circular patterns, like you can see here, if you put epithelial cells after a certain density, the cells start to collectively move and rotate around these discs. But also, if you use other kinds of patterns, like ring-like structures, like you can see on the right, you can see that once the cells feel the rings, they start to collectively move and in a cohesive manner and for a very long time. And so in these kind of movies, we can do some tracking of the cells and measure different kind of aspects like the speed or the forces that the cells might apply on the substrate by using MATLAB or other tools that are available. But of course, the interest of studying collective cell rotation also occurs because it happens in vivo. And maybe the most well-known example is the Drosophila follicle epithelial rotation during the Drosophila egg chamber elongation. And as you can see here, these green cells here rotate around the egg, which is a very interesting phenomenon. We basically still don't really know exactly why it's happening, but it's believed that this collective rotation helps the egg to get shaped. And recently we also saw rotation in cancer cells exiting some spheroids. So again, we go back to in vitro studies here, but these cells, when they exit the big spheroid, sometimes they get clogged inside the collagen gel, and we can appreciate some collective cell rotation around their main axis, which may allow them or facilitate them to invade more. And so in order to recapitulate these experiments, one of the first things we did was to put single MDCK cells and grow them inside 3D Matrigel gels. And as you can see, after a couple of days inside this Matrigel, they form multicellular cysts or acini that are usually spherical, that you can see in this 3D representation here. They're really round. And if you look inside them, you can see that they're basically hollow, and they form very nice monolayers with a very strong actin apical staining here, and inside basically they're empty. Now, if you block cell division in these spheroids, you will see that basically you will start to have collective rotation of these cells inside this Matrigel gels, which is very exciting for us to see, because basically this collective rotation in the Drosophila egg chamber happens also in low proliferative periods. Now, the very interesting part was that we didn't see only formation of spherical cysts, but also tubular cysts, like this one, for example. And as you can, I hope, appreciate, we could see collective cell rotation over time for long periods of time, like hours until 20 hours. We were able to fix and stain these tubes and be sure that they have this tubular structure, and we're able to look inside. We can see the collagen 4 staining for the Matrigel outside, the fact that these tubular structures are hollow, and we can also measure some speed. So if we look at the Y and X axis from this movie, if you measure the speed of the cells just by tracking the nuclei on the Y axis, the longitudinal axis, you will see that the speed are pretty much zero over time, because the cells move a bit randomly up or down. But if you look in the rotational velocity, so the velocity of the cells in the x-axis, you can see that it's constantly positive in this case, meaning that the cells constantly move towards the right, so they have a rotational phenotype. Now, we wanted to go deeper into that study and have more detailed information about the rotational dynamics. So we decided to use another model. And so we went to fixed PDMS tubes that we can produce in the lab, basically. And this is what you can see here. We can have tubes, hollow PDMS tubes of different diameters, ranging from 25 up to 250 micrometer, which is the range of physiological tubes that we can find in the organisms. And basically, the way that we do in the experiments that we have these PDMS blocks in which we have these hollow tubes. We put our cells on both sides of these blocks and we let them grow. And once they become confluent, they start entering inside the tubes. And so we can follow their migration. And so this is how it looks like. Basically, you can see here the histone GFP cells inside a 50 micrometer diameter tube. We can see that the tubes are hollow, so the cells really populate the walls of the tube. So you have a clear lumen inside, and you can also have stainings of different kind of proteins of membrane and different parts, so we can really appreciate that lumen formation. And we can appreciate rotation. So this is an example of a 50 micrometer tube. You can see the cells, they are advancing, already rotating, and then they meet, and you will see that once they coordinate, they start all together to rotate towards the right. This is one example for 50 micrometer, but we also see rotation in larger tubes. This is an example of 100 micrometer. You can see in this case, they rotate towards the right. In the other case, we're rotating towards the left. I'm not going to go into details, but there's no clear chirality. Basically, it's random if they move right or left. And so in order to study in detail the dynamics of these movements, we process these 3D confocal movies in image J by just opening the tube. So basically, from a 3D tube here, we open the tube and we get an X and Y image, a 2D image, in which it's much easier to do any counting. And so we end up with something like this. So these are open tube images of tubes from 25 up to 150 micrometer. And we can track the nuclei movement. And as you can see, these trajectories, each line here represent the movement of one nuclei. You can see that over time, this is the time here, these nuclei move all towards the same direction in the parallel lines. You don't have much exchange of neighbors, and it shows that basically you have a collective rotational movement. And if you look at the velocities, you can see that there is an increase in rotational velocity from 25 micrometer diameter tubes up to 100 micrometer diameter tubes. We have the peak velocity, and then the velocity starts to drop down again at larger diameters. And if you look at 200 micrometer or 250 micrometer diameters, very, very large tubes, then you lose this collective cell behavior. And one hypothesis is the fact that maybe the cells do not sense the curvature anymore because the tube is too large. And so then we went a little bit more into the biology of the system. First, we wanted to check if during this rotation, the cells actively migrate, if they're polarized. And so we did an experiment using histone 1 GFP cells mixed with YFP PBD cells, where we have where we can see the fluorescence of the active rock, which localizes at the laminipodia of the cell during migration. And as you can see here in this fire staining, we can see that the cells, as they migrate, they really are polarized. So you can see these cells here, and they rotate towards the right. Again, this is an open tube conformation, and we can clearly see the polarity of the cells as they move collectively during their rotation. Now, this polarization of the cell is very important, because if you block this polarity, if you using a RAC inhibitor, if you block the ability of the cell to form laminipodia, the cells start to collectively rotate and just randomly move. Now, we wanted to see if the actin cytoskeleton is different, because, okay, we know that the cells polarize, but how do the rest of the actin cytoskeleton looks like? So basically, we fix the cells during rotation and stain for actin. So this is one movie. You can see the cells are rotating towards the right, and then we fix that specific moment, and we just stain with phalloidin, and we can see that the actin fibers were really perpendicular to the direction of the motion, which was quite surprising, because we would expect that the actin fibers were more parallel to the direction of the rotation, but it was actually perpendicular. And this is true in all the different kinds of tubes that we tested, from 25 up to 150. And you can see that in the larger tubes, you start to have a shift of the actin fibers towards the direction of motion, probably because the curvature becomes the tube becomes too high. And one of the last things that I'm going to talk about is just the traction force microscopy that we tried to do in these tubes. So we were able to fabricate soft tubes of 30 kilopascal, in which we were able to put bits. and we can spot the rotation of the cells and measure the force that the cells exert on their substrate by just calculating the displacement of these beads. And what you can see in this graph is that surprisingly, when the cells do not rotate, when they randomly move, the magnitude of the force, which is represented here by this yellow line, is quite big. But once the cells start to collectively rotate, the magnitude of the force decreases. And we couple here with a rotational velocity. So you can see when the rotational velocity is close to zero, the magnitude of the force exerted on the substrate is quite high. But once the rotational velocity becomes positive, meaning that the cells start to rotate, the magnitude of the force kind of decreases. So that's a very interesting result, because we're kind of expecting to see the other way around. And basically in this heat map, you can see this thing that once rotation starts, the traction force kind of decreases. And just to finish, I want to say that we're also looking at other kinds of curvatures. So all these experiments that I've showed you so far were in a negative curvature, so inside a tube. But we're also looking at positive curvatures. So what's happening when you have an epithelium of cells on top of a fiber? And so basically it looks like the same thing. And surprisingly, we also see collective cell rotation, even if you have this positive curvature. So you can see we do the same experiments. We follow the trajectory of the cells over time. We can see that they collectively rotate. The rotational speeds are similar, that the ones we see in the negative curvature. But in this case, the actin fibers orient completely on the opposite side. They follow the rotation direction, basically. And this is true for, again, all the different diameters that we have seen. So basically, we can say, with the result that we have so far, that the big, large actin fibers of the cells orient themselves according to curvature, but these do not change the fact that the cells can still collectively find an homogeneous and coordinated way to move all together. So I'm going to stop here. I would like to thank my team for all the support and different people who have helped me with using MATLAB and other tools that I didn't know how to use before in order to have this result so far, and the imaging facility, and of course you and the organizers. Thank you.
Thank you, Alexandros, for the talk. Any question? Elena Canver, go ahead.
Yeah, hi. Can you hear me?
Yes.
Yes.
Okay, so very interesting. I have two questions. First, have you tried to look at other components of the cytoskeleton, like microtubules and so on and so forth?
So not yet, no. We want to look at actin because we will basically try to focus on the actin cytoskeleton. Mm-hmm. But we want to look at the microtubules as well. It's more complicated because actually the staining inside these tubes, the practical experimental procedure is a bit complicated. It's not easy. It's like a 3D staining, but it gets tricky sometimes. So phalloidin is easy to make, as beta-catenin and other proteins also, but the microtubules is a bit more tricky. So we are still working on that.
Okay, and second question, and I'm thinking about it might be way too complicated, but have you tried to see the cells on other shapes, more complicated ones, in which the curvature can change, like a torus or something like that? Is it too complicated to make simply?
Yeah, that's a very interesting question. We get that a lot, actually. It is quite complicated to make. To make these tubes, we have some fibers in which we embed inside the PDMS, which is still liquid, and then we bake it, and then we remove the fiber, and we are able to have the tubes. But if you want to do more complicated shapes, it gets really complicated. So we haven't tried so far to do very, very complicated shapes. We wanted to have tubes that change their diameter, like you have the same tube with different diameters, but we haven't even realized to do that yet. So, yeah.
Okay, thank you.
Thank you.
Okay, so next we have a question in the chat from Juan. Do you have any idea on the mechanism? Is it related to adhesion proteins or to curvature sensing proteins?
So actually, we don't really know. We believe that actually the cells sense the curvature and adapt their cytoskeleton to the curvature, but maintain their ability to collectively move. And I think that this is mostly related to the fact that the cells are really well connected to one another. So the adhesions are very strong. I didn't have time to show you here the data, but we show that if we use EGTA or other drugs that you can disturb cell-cell adhesion, you completely lose this collective movement. And also the fact that the adhesions are reinforced in the longitudinal axis of the tube, meaning that there is a very strong connection longitudinally, which allows the cells to collectively move. But as for the mechanism, unfortunately, we don't have any more clues on that yet.
Okay. Just so maybe a question, did you try to reduce, for example, cell-cell adhesion?
So we tried, yeah, in a sense. We used EGTA, which completely disturbs calcium-dependent cell-cell adhesions. And in this case, you break, the cells stop to rotate collectively. And it's very interesting because you add the drug, the cells stop to rotate, and then you remove the drug, and then you see that after a couple of hours, they start to rotate again. So it's very consistent, actually. And it's funny because sometimes collectively the cells rotate towards the right, then you add the drug, you remove it, and then the cells start to rotate on the other side, on the left. But if you just remove one part, for example, if you use, we have used ECA-adhering knockout cells. And in this case, the cells still could rotate because there are other proteins that can, let's say, compensate for the loss of one protein.
Okay, thank you. So there is also a question in the chat. How do you explain the difference in actin filaments orientation between fibers and tube?
Yeah, so I believe that is the easiest answer, let's say, to that question would be that the fact the cells sense the curvature. And if you look, if you imagine a cell on a negative curvature, it's more difficult for the fiber to be hanging, basically, and not touching the substrate. Whereas if the fiber is oriented in the longitudinal axis of the tube, it can always be touching the substrate. So I think it's a matter of that on a positive curvature, it's easier for the cell to have the fibers oriented towards the direction of migration, which also helps the migration. But on the negative curvature inside the tube, it's easier for the cell to align the fibers, the actin fibers, with the longitudinal axis. And basically, I think it just helps the cell to really adhere. And the idea that we have so far is that basically the large actin fibers, their role is more to connect the cells with each other, because we can see actually the actin fibers ranging to many cells. You can follow them in many cells. You cannot be sure that it's the same actin fiber, but it really seems that they connect many cells. And we believe that their role is more to keep the cells together and assure this collective movement, rather than actually being used for the actual rotational migrational motion, which there is the different actin network. It's more the lamellipodia actin network, which is different, it's more branched. Yeah.
Thank you. So maybe one last question from Jacqueline, actually. She says, great talk. Is there any differentiation that coincides with movement?
Differentiation, I don't think I understand the question.
I was just wondering if, like, because these cells are being cultured for a while, right? And there are studies in mesenchymal cells studying cells on different curvatures that they have a really different gene expression. So I'm wondering, do you see any, have you looked at all for any kinds of suggestion of a differentiation event, or at least a fate transition that might help reinforce these movements?
No, we haven't looked at that. That's an interesting question, actually. We haven't looked at that at all. No, it's true that we're using MDCK cells that have been cultured for a long time, and I'm sure they're really modified compared to other cells that you might think. But we want to actually use different cell types to see if we can see this effect on different cell types, and maybe that could answer this question. Yeah. Thank you.
Okay. So thank you, Alexandros. So we'll keep other questions for the break at the end of the session. So thanks again for the great talk. And now, Xiu, you can begin to share your screen. So next